Hello. Thank you for the organisers for inviting me to speak to you today, and I can only apologise for not being with you in person. I will give you an overview of the UK Crop Microbiome Biobank, approaches to cryopreservation and data curation. So just to give you a little bit of introduction about myself, I am a Senior Research Lead of Biological Resources at CABI and am a microbiologist by training. I currently lead the UK Crop Microbiome Cryobank and work on several other projects, including two EU projects, the UK Crop Microbiome Cryobank and the European Microbiobanking Enabler, as well as contribute to the Research Centre for the Holobiont for the Leverhulme Foundation. I hold several honorary roles, including being a board member of the International Alliance for Phytobiomes Research, and I'm an international representative to the United States Culture Collection Network, amongst other things. In my talk today, I'll give you a quick overview of CABI and what we are about as an organisation. I will then look at the history and context of microbial biobanking and ask the question, is biobanking for microbiomes needed? I will talk about the Phytobiomes vision before giving you a little more detail about the project that is the UK Crop Microbiome Cryobank, before going on to mention the importance of international networking and research collaborations that we are achieving through the European Microbiome Biobanking Project and EU Microclimate.
CABI is a UN treaty-level international not-for-profit organisation owned by 48 member countries. It has a global reach of some 500 staff, and our key aims are to address global concerns such as food security and food safety through research and international development cooperation. Our areas of expertise include crop health, which include increasing yields, particularly in smallholder environments, development, communication and extension, digital development, the control and management of invasive species, publishing of abstracts and journals, and also value chains and trade. We are a global organisation. We are headquartered in the UK, but have CABI centres in Switzerland, Trinidad, Brazil, Ghana, Zambia, Kenya, Pakistan, India, China, and Malaysia. CABI itself hosts several key biological culture collections. Our collection was founded in 1947, but has since expanded to cover the UK National Fungus Collection, the British Antarctic Survey Culture Collection, the UK National Collection of Wood-rotting Fungi, and a small collection of Phytophthora and Pythium known as the Aquatic Phycomyces Collection. We recently founded the BBSRC Crop Microbiome Cryobank, and more of that a little later.
We should start by considering the status of culture collections internationally, or the state of the art. Historically, culture collections store exenic cultures, that is, cultures in pure format that can be cultured, maintained and distributed. Only very rarely do culture collections store microbes in anything more complex than one or two organisms as part of a consortia. So adding the microbiome on top of this has another area of complexity. We often see that human and animal microbes are often prioritized over environmental and plant-associated organisms. This differs somewhat to biobanks. Biobanks tend to be reference collections of dead or fixed material, such as tissue banks and museum collections. Although, under this remit, you can also include viable seed banks or medically related biobanks, such as IVF clinics and blood banks. It is fair to comment that these designations are not mutually exclusive. in that some culture collections will store fixed and preserved material, and as I have alluded to, biobanks may incorporate living material. But I guess the take-home message is it's time for everybody to work together. An example of a culture collection that supports traditional microbiology is a collection that I was very honoured to curate myself for over 10 years, and that's the Kabi IMI collection. It was founded in its current form in 1947 by Major Dade when it attained national status. That was when the UK decided to reorganise their culture collections, with Kabi becoming owners and custodians of the national fungal resources. The collection for many years was known as the International Mycological Institute Collection, and we retain that acronym today for our strain accessions. The collection itself consists of over 30,000 living microbes, DNA and associated data, representing some 6,000 species. Of these, 28,000 are fungi and around 2,000 are bacteria. And they come from across the agricultural world and the environment. Over 55% of these strains are plant associated. Given the history of Kabi and our makeup of 48 member countries, it is therefore unsurprising that the collection has a global distribution. The cultures are maintained using state-of-the-art technologies, such as cryopreservation and freeze drying. Kabi was one of the first culture collections in the world to routinely use both cryopreservation and lyophilization, techniques that have been in use since the late 1960s. We also incorporate other collections, as I've mentioned, and importantly, we are fully compliant to the Nagoya Protocol of the CBD.
But that's okay for microbes in pure form. But we know microbes are everywhere in, for example, the food system. They're in the sea, they're in rivers, they're in water, they're underneath our feet in the soil, they're in the air. Microbes are an important part of everything we do. But in these environments, they tend to live as communities of many thousands, if not millions, of other microbes interacting with their hosts. This was a slide, an infographic, produced by the very successful Horizon Europe Microbiome Support Project. But it shows the relationships of microbes with the wider environment. One concept to consider is the phytobiomes concept. This slide produced by the Phytobiomes Alliance, an international not-for-profit industry academic collaborative. It considers not only the microbes, but the relationship between the plant, the microbes and macroorganisms, including macrofauna, animals and other animals and plants, soil, and that is all influenced by the weather, water and climate, and influenced by management practices. The systems-based approach is how we should look at all research in the area of microbiome.
So, with that in mind, how do we begin to underpin research in the microbiome? In this case, the crop, plant, soil microbiome. And so, in 2021, during the COVID pandemic, we established the UK Crop Microbiome Cryobank. The aim of this project is to establish a cryopreserved and characterised crop microbiome resource. Importantly, to establish baseline characterizations and the metagenomic analysis of all the samples we intended to hold. The aim of this is to underpin UK and international crop research which builds on existing UK activities. Our main deliverable is to provide a comprehensive platform to facilitate research towards optimizing plant yield in an integrated crop management framework. This will then provide an infrastructural baseline for other project and science-based activities. The project focuses on key UK crops, including wheat, oilseed rape, otherwise known as canola, barley, oats, sugar beet, and fava beans. The project is a collaboration of 5 key UK crop and soil science research institutes. These include the John Innes Centre, the James Hutton Institute, Rothamsted Research, CABI, and SRUC, which is Scotland's Rural College. Collaboration is key to the success of the project, and the project is centred around 4 key work packages, each overseen by one of the collaborative partners. Work Package 1 involves the collection, production and culture of all downstream samples used in the project. SRUC and the James Hutton Institute then undertake all of the genomics and the bioinformatic analysis of all samples. The physical samples are located by cryopreservation at CABI as part of Work Package 3. And then in Work Package 4, the John Innes Centre are developing utility from the investigation, that is, looking at the samples to see where they can be used for interventions in agriculture. I will provide a bit more about all those activities in the next slides, including Microbiome Base, the web-based platform that we are using for the project. The most important work package, because without it the project would have had no samples to work with, was Work Package 1. This was overseen by Rothamsted Research. This involved collecting bulk soil from across the UK, from Scotland in the north, right the way down to north of London in the south. 3 soil types were collected, representing 3 distinct textural classes, which was sand, clay, and silt. The bulk soil was then processed at Rothamsted, with aliquots and subsamples sent to CABI for storage. The bulk soil was also fully genomically characterised. The interesting part of the experiment was then a large pot-based experiment where we grew the 6 different crops in the bulk soils. At the end of the experiments, the plants were harvested, with the microbiome samples of the rhizoplane sent to CABI, along with 36,000 isolates that were actively cultured from the soil and the resulting experiment. The second work package, which was overseen by the Scotland's Rural College and the James Hutton Institute, was equally important. It was very important at the start of the project to work out exactly how we were going to sequence and characterise the project samples. With a finite resource, we had to make sure it was done correctly and in a standardised manner. As mentioned on the previous slide, we have analysed both bulk soil and rhizosphere samples, but also bacterial and fungal isolates. We have used 16S and ITS amplicon approaches, as well as shotgun metagenomic sequencing, with some strains selected for whole genome sequencing. All sequence information is publicly available, and we have established a workflow to utilize biosamples of the European Nucleotide Archive EBI. All of this is then stored on mGenify, EBI's microbiome analysis platform. And all data is accessible through Ag Microbiome Base, which I will show you in a few slides' time. And now I'm going to talk about task 3, which is the work package led by myself, which is the cryopreservation of all project resources. And I'm going to go into a little bit more detail about our approaches to cryopreservation.
But there are challenges of preserving a complex community. And when I'm talking about a complex community, I'm talking about more than 2 or 3 organisms, potentially up to many hundreds of organisms. And that applies to microbiomes. Douglas Adams, who wrote The Hitchhiker's Guide to the Galaxy, came up with an interesting hypothesis. If you try and take a cat apart to see how it works, the first thing you have on your hands is a non-working cat. This was taken a little bit further by a collaborator at Imperial College, Thomas Bell, when he said discovering which part of the community can be taken away without altering functioning would tell us about which components affect community structure, what are important, and how then do we differentiate between a heart, very important, and a whisker, equally as important. And so when we're preserving microbiomes, we have to consider the integrity of our endpoint. Our preservation method of choice is cryopreservation. That is storage at ultra-low temperature in a cryogenic gas or liquid, typically at temperatures of around -180 degrees or below. We do not consider storage at -80 to be a suitable method of preservation. The method itself has been used since the 1960s and is seen as the Rolls-Royce or the best method of storing not only xenic cells but now also microbiome-based material. You need to ensure a careful approach to ensure sample integrity is not compromised. And our key aim is to preserve not only the taxonomic and physical structure of the cells, but also the functional potential of the communities that you're preserving. When using cryopreservation, you have to be very careful not to damage your samples. As the cooling curve shows on the left of the screen, when you cool to below freezing, you actually get heat generated. If this step is not controlled, then it can cause irreversible damage until you can get the temperature low enough where the eutectic solution actually freezes, and you can actually control the process in much more greater detail. The diagram on the right of the screen shows that at every stage of cryopreservation, not just the cooling process itself, you can see damage occurring, whether it's during the preparation, the addition of cryoprotectants, or after freezing on the thawing recovery. The prime aim here is to avoid cryo injury. This is not only important for cells that are kept in pure format. When dealing with populations, it's of more importance because you don't want to alter the population of the cells you're trying to preserve. For the cryobank, we use 2 methods of cryopreservation. Firstly, a control rate cooling method using a Stirling cryocycler. And we use this to capture most freeze-tolerant organisms. We also use an encapsulation vitrification approach to capture more delicate and freeze-recalcitrant organisms. This particular method involves immobilizing cells in calcium alginate, and then you remove the water using a controlled dehydration stage. You get to a point where the cell has less water, and so when you freeze it, you get less ice damage. What we have also learned is never to freeze dried soil samples or microbiome samples, and more of that in a little bit.
So what have we actually stored? Well, I'm pleased to say that over the course of a 4-year project, we've amassed nearly 40,000 individual samples. These consist of 4,800 individual cryo vials preserved using Stirling cycle control rate cooling and encapsulation dehydration. They are the microbiome samples consisting of soil and rhizoplane. And in addition to that, we've got 36,000 isolates in microtiter plates. All samples have been fully conserved, and you can find details of them in Ag Microbiome Base.
So we've preserved all these samples, and the key question you may be asking, well, do they survive? Do your methods work? I'm pleased to say for the 36,000 isolates, we have over 95% viability, which is exceptional for any culture collection. And we're working at the moment on fully characterising those organisms, which will help with our understanding of the biological questions that we can ask of such resources. But what about the soil samples? Well, we've looked at the rhizosphere of Brassica napus, oilseed rape or canola, as you know it, and we compared 3 methods of cryopreservation: freeze drying, control rate cooling, and plunge cooling, against a range of control samples. Amongst the methods we've used to analyse the functional potential of what we've preserved, we have used Biolog ecoplates.
In this study with the Imperial College, where we looked at the effectiveness of our preservation regime using Biolog and enzyme and ATP assays, we found some really interesting results. Particularly that plunge cooling in liquid nitrogen and control rate cooling were repeatedly found to conserve microbial functionality almost equally. You can see this in graphs B and D. Interestingly, if you look at graph C, which is samples that have been freeze-dried, we see a total loss of functional activity. These results are still preliminary. We have more analysis to do, but what it does show is that our cooling approaches are safer than more sporadic approaches such as freeze drying. All information about the UK Crop Microbiome Cryobank is available on our project website, www.agmicrobiomebase.org. Here you can find information about the partners, the team, our resources, and also key contact points. Interestingly, and perhaps most importantly, it's where you'll also find our data resource. If you select the Data tab on Ag Microbiome Base, you will find our Ag Microbiome Data Explorer. Here you will find our complete characterized dataset, which is fully searchable, and you can also download it to interrogate the data yourself. You have the ability to undertake bespoke searches by selecting any of the fields based on the soil ID, the crop, or the geographic region within the UK. The main USP of Ag Microbiome Base is the fact that we link not only the soil physical parameters and that massive data set, we link that with the experimental details of the project, the physical sample itself, the metadata and genomics information, and importantly, we maintain the link to the EBI resources that are also publicly available. At the current time, we believe this is the only database anywhere in the world that does all of those things. We're particularly grateful for feedback, so if anybody present today would like to try and experiment with the database, please let us know how you get on. We'll be grateful to hear from you. Finally,
In Task 4, we will demonstrate the utility of the resource. We have established a pipeline that will allow us to design syncoms based on their phenotypic and genotypic properties. These will then be tested in 2 stages, a pilot test and then a secondary test that will allow us to upscale to potential application. The picture on the screen shows an EcoFab1 device, and that is a Bardley seedling. EcoFabs allow us to look at syncoms and their effectiveness, and were developed by Trent Northern in the States. Our initial funding runs out at the end of this year, so we are now collating results, writing papers, producing protocols, so please watch our website for further announcements. And therefore we are now looking at the future of the resource. CABI has undertaken to continue the resource as part of its collections, but we'd like to attract other funding, whether it's through expanding the resource to cover other crop types and to make the resource accessible for public accession. Key to this is international collaborations, as we believe the Cryopreservation model is a utility for other systems worldwide. But most importantly, it's the future of One Health-based resources and the ability to answer key biological questions. As our project has shown, it is only when you have the resource, the data, and all of the key components that you are able to answer key questions of your data, which is very important in ensuring that resources are usable and sustainable into the future. Another important project that we're involved with at CABI is the EU Microbiome Biobanking Enabler Project, EU Micro for short. RI stands for Research Infrastructure. Partners include the Austrian Institute of Technology, who are the leads, along with DSMZ in Germany, EMBL, IMRE in France, MUG, which is the Medical University of Graz, and Sorbonne University and their Roscoff Environmental Marine Station. The project seeks to establish an operational blueprint for the very establishment of microbiome biobanking. That means the methods and technologies that we're looking to develop. Ultimately, it seeks to transfer these to end users, but also to ensure translation with other environments. The project is composed of a number of complementary work packages. I am fortunate to lead the work package on development of cryopreservation procedures, but there are supplementary work packages on keystone species and ecology, the Nagoya Protocol and regulation, the need to establish data infrastructures for microbiome biobanking, standards, and a legal framework. As I mentioned in the previous slide, a key focus is translation to end users and improving the TRL, or Technology Readiness Level. The title of the work package I lead is Validated Protocols for Preservation and Propagation of Complex Microbiome Samples. There are 4 main aims of this work package. These include developing new preservation approaches for microbiomes and syncoms, development of sampling procedures and the use of stabilization reagents that don't require the use of cooling chains, that is, the use of cryogenic gases, the development of novel propagation technologies, and approaches for optimized assessment of microbiome functions. We have undertaken 3 experiments, one focusing on soil, the other seeds, and the third looking at marine systems. The first stage involved testing existing preservation protocols.
Our initial results are incredibly encouraging and has involved the development of a cross-institute experiment, which has been very complex to establish and develop. We will be using the outputs of this research to inform further method development and optimization in the latter stages of the project. It is clear that the experiment would not have been possible without the collaboration and experience of institutes from across the European Union.
So in summary, what are the next steps? There's clearly a need for broader regional and international infrastructure for microbiome biobanking. The UK Crop Microbiome Cryobank provides a model of an agri-focused microbiome resource. Curated resources allow data scrutinising and the ability to ask complex biological questions. Further work is required, be it through the optimisation of the preservation approaches or research relating to the retention of functional potential. Ultimately, biobanks need to be there to support the biological communities and research communities that they support. And that can be through patent deposits, environmental deposits for strange release into the environment, or for such things as the Nagoya Protocol. The engagement of industry in all of this is essential. We share common challenges.
None of the research that I've spoken about today would be possible without our amazing team of collaborators. For the UK Crop Microbiome Cryobank, it includes Tim Morklin, Jacob Malone, Kat Thompson, Ian Clark, Nick Holden, Sue Jones, Rodrigo Tate-Tani and Peyton Yao. In Europe, we are grateful for the support of the Microbiome Support Consortium and also the EU Microbiome Biobanking Enabler Team, including Perrine Portier, Michael Schloter, Tanya Kostic, Rob Finn, Jorg Overman, Cornelia Stumpfer, Ian Provert and their teams, as well as our colleagues at Imperial College, Professor Tom Bell and Tom Lilly, and not forgetting my own collaborators and team at CABI, including for the Cryobank, Helen Stewart and Miguel Bonin. It only remains for me now to say thank you for your attention. We will welcome any feedback you can give us, so please do reach out. We are very, very open to discuss avenues for future international collaboration, something that CABI as an organisation has stood for many, many years. Thank you.